So the learning outcome for today's lesson is GMO in agriculture is to explain that genetic engineering may help to solve the global demand for food, to improve the quality and the productivity of farmed animals and crop plant. So productivity also means quantity, or I prefer to use the word yield. Okay, so you want to improve quality and quantity of the plant and animal, okay, or quality and the yield of the plant and animal. So the three examples you're going to look at is GM salmon, herbicide resistant in soybean, and insect resistant in cotton. Then after that, we also have to discuss, because all these GMO, right, contain a new gene, and a foreign gene has been inserted into it, which will not be there in a natural environment, in a natural way. So when we introduce this GMO to the environment, we have to think about the ethical and social implications. That means, is it right for us to introduce it? Then we have to consider, how will it affect the environment? How will it affect other organisms in the environment? How will it affect us as consumers? So every time when we say ethical and social implication, we have to look at both the pros and cons of introducing this GMO, and therefore, should we continue to introduce them and grow them or not? Okay, so this is called ethical and social implications. So the three GMO they're going to learn is A, fast-growing GM salmon, salmon that grows very fast. B, herbicide-resistant soybean. When you spray herbicide, all plants will die, only this soybean will survive. C, insect-resistant cotton. That means when the insect feed on the cotton, the insect die. Okay, GMO stands for genetically modified organism. Okay, you look at this diagram. These two are salmon fish. This is GM salmon at the back, and in front is the farm salmon. Now, both fish are 18 months old. Can you see how come this fish 18 months old so big and this one so small? It's because this GM salmon can grow very fast. It can grow very, very fast, so that within 18 months it's already this size. Now, conventional salmon fish, usually they only grow during spring and summer. So in one whole year, they only grow 6 months. Whereas GM salmon can grow the whole year throughout. So because of that, GM salmon, right, we only grow them for 1.5 years only. Whereas the normal salmon, we need to grow them for 3 years to reach the marketable size to sell. So this one will reduce the number of the timing by half, because 1.5 years this size, we can already sell the GM fish, but the normal fish is still too small, right? So can you see how powerful is the GMO, right? How powerful it is to create the GMO. Right, so what happened to this GMO? How come can grow so fast? Because we have inserted a gene called the growth hormone regulatory gene to promote growth of the salmon. And then to the upstream of the gene, we have inserted a promoter, which the scientists have obtained from ocean pulp, which grow the whole year. So that when this promoter is inserted to the upstream of the gene, this gene is switched on the whole year. So the fish will grow the whole year, have become very big. So what is a gene delivery method? Micro injection. So they use a small, tiny little pipette to inject the gene plus its promoter into a fertilized egg, to the nucleus of the fertilized egg. So after that, when the cells divide, all the cells of this fish will contain this inserted gene, and they can grow very fast throughout the whole year. Now, gene technology is very advanced in US and Canada. So this GM Atlantic salmon has been developed in US and Canada. It can grow throughout the year instead of just spring and summer. But because it can grow throughout the year, it will need more food compared to the wild salmon. Wild salmon means the natural type of, okay, that you can find in the nature. It's called wild. But it will reach marketable size in 18 months versus 3 years. Now you see, it can grow very fast, but because it grows very fast, you need to supply them with a lot of food. So they need a lot of food. So if they happen to escape to the wild, say for example, you're growing in your agriculture tank here, okay? And then there's a little river nearby. Don't know, somehow or rather, this fish escaped to the wild. Now, our main concern is what happened when it escaped to the wild? Can it contaminate the wild species, the wild salmon or not? Contaminate means, will this gene be transferred by fertilization when the mating occurs? So will it be transferred through mating to the next generation? Because we learned that whenever we use genetic technology, we do not want the new gene that we produce, we insert, to be transferred to the next generation. So now our concern is, if the fish escape, will it mate with the salmon fish in the wild and then produce a hybrid offspring which carry the introduced gene or not? Then this will actually pose a problem in evolution. That means we will play God already, we will change the genetic makeup and the phenotype organism already. So we don't want that to happen. So the solution is, yes, they may escape, but if they escape, they cannot grow and they cannot survive because they need a lot of food, right? They grow very fast, they need a lot of food, but in the wild, in the natural environment, they may not be able to get that much food. So even if they escape, they may not survive. Okay. So what is the ethical consideration for this type of organism? We want to avoid contamination of wild species with this particular gene through crossbreeding. So some scientists say, since this is your concern, right, escape to the wild and contaminate wild species, they propose you only rear sterile females. So you make sure that the salmon fish that we rear, the females are all sterile, so they cannot lay eggs. Okay, and also propose to farm them in land-based tank. So farm them in the land rather than in the river where it may escape easily if there's a leak in the tank, okay, or in the net. So GM Atlantic salmon is less capable to compete with wild salmon in the natural environment. Even if they escape because they need a lot of food, they cannot get enough food, they will die. So they cannot compete with wild salmon. So even if they escape, there's not a problem. US Food and Drug Administration, FDA, has declared that GM Atlantic salmon are highly unlikely to have any significant effects on the environment. Because of this, okay, this is a problem, potential problem. So after that, they look for potential solution. And then, is the problem possible or not? Here, they say no, very unlikely, and it is as safe, as good as conventional Atlantic salmon. So they say that both are similar. Even if they escape, they cannot contaminate, so it's okay to eat. Okay. Okay, so on the left, you see a lot of eggs, right? Salmon eggs. So after development of the GMO, in the year 2013, Canada allows GM salmon eggs to be used to grow new salmon. In 2016, Canada allowed GM salmon to be sold to the consumer like us. In 2019, US FDA allows GM salmon eggs to be used to grow salmon. So this is to show you that even though the genetic engineering technique has been developed many, many years ago, it takes many years to actually develop the technique, produce a product that is safe enough to be marketed. So it takes a lot of research and development in order to get the final product. Okay. Okay, so that's for GM salmon fish. Now we want to look at GM soybean that is herbicide resistant. So this is the soybean inside. So this is the bean, inside is the soybean. and this is the plant. So how to make the herbicide-resistant soybean? Why soybean? Because soybeans are used as a food source for animal, for human. Is used as biodiesel, means we use it to provide us with energy, just like we use diesel. We burn the diesel and provide us energy. So the top producer for soya bean are US, Brazil, China, India, Canada, Ukraine, and so on. Okay. So it's needed in the whole world because we feed on what? For us, we eat bean curd, tofu. Tofu is made of soybean. We drink soy milk, it's made of soybean. Okay, so a lot of food, a lot of consumers that feed on soybean. So it's important to improve its quality and yield. So what is it scientists propose to modify? They want to make a transgenic crop plant. Now, transgenic crop plant is a word for a crop plant that has received a foreign gene, just like transformed bacteria has received the foreign gene. Transgenic crop plant has received the foreign gene. So we want to make this plant not able to be killed by the herbicide, so that we can spray the entire field with the herbicide, and this herbicide will kill all other plants, which we call the weeds. Weeds are unwanted plants growing in the field or in our garden. Okay, weeds. Any plant that you don't want them to grow, but they grow, we call them the weeds. So we want to spray herbicide to kill all the weeds, but only our transgenic crop plant cannot be killed by the herbicide. So therefore, when you kill all the weeds, it will reduce competition of the weeds with our crop plant for space, for light, for water and ions. So they can get more sunlight, they can get enough water and mineral ions from the soil, so they can grow better, so the yield becomes higher. That's what we want. So how does the herbicide work to kill the weeds? So if you look at this pathway, this is a pathway in the synthesis of 3 very important amino acids: the tryptophan, phenylalanine, and thyroxine. So these are amino acids that are very important, and these amino acids are needed for the synthesis of protein. Right? Amino acids are needed for synthesis of protein, and these are needed for growth of the plant. Okay, so when the farmers spray the herbicide, the active, now the name of the herbicide is called Roundup, commercial name, but actually the active ingredient is called the glyphosate. So this chemical glyphosate actually inhibits an enzyme in the pathway. So when this enzyme is inhibited, this pathway cannot, this reaction cannot occur. So all these amino acids cannot be formed. So when it cannot be formed, there's no protein that being synthesized, so the plant cannot grow, so it will die. So for all the wheat plant, they'll die because of this. But for the herbicide-resistant plant, what happened is that we have inserted a special gene which helps to code for the enzymes to give an alternative pathway to the synthesis of these 3 amino acids. So now, because of the genetic engineering, this gene is inserted to the plant, so the plant becomes herbicide resistant. Herbicide resistant, correct? So why is it resistant? Because now they got this enzyme that can allow them to synthesize this amino acid. So even though the scientists, the farmers spray the herbicide, it will not affect this enzyme. So the plant will continue to produce amino acid proteins and continue to grow. Just like here, spray herbicide, herbicide-resistant plant grow because they can produce protein. Normal plant cannot grow, so they cannot produce protein, they die. So these are the weeds. These are the herbicide-resistant plant. So eventually wheat die, our crop plant grows. Okay, so why? Because herbicide glyphosate, when sprayed on a plant, is absorbed in the leaves, transported to the plant body. And in the cells, it inhibits enzyme for synthesis of 3 amino acids, phenylalanine, tyrosine, and tryptophan. So that will prevent growth at the growing tips. Because when a plant grows, it is the shoot tip, root tip, branch tips that grow. So the cells there cannot synthesize amino acids, cannot grow. So the plant will die. So see, wheat plants die, the herbicide resistant one grow. Wheat plant die, herbicide resistant plant grow. Okay. So what is the gene we've introduced? The gene is a gene coding for enzyme that catalyze the synthesis of 3 amino acids. And this gene comes from a soy bacterium. And it is being introduced into the plant cell using these 2 vectors, Ti plasmid and the bacteria Agrobacterium tumescens. Now, why is it called Ti plasmid? Now, Ti plasmid naturally is found in this bacteria. So that's why you want to transfer the gene to this Ti plasmid and transfer this recombinant DNA into the bacteria. Then use this bacteria to transfer the gene into the plant cell, because this bacteria naturally infect the plant cell. Okay. Now, Ti stands for tumor inducing. Why is it called tumor inducing plasmid? Because Agrobacterium, agro means agriculture bacterium, are bacteriums found in the soil. And it's also called tumor faciens, because this bacteria, this soil bacteria naturally infect the cells in the wound of the root of the plant, and then it can cause tumor formation in that area. So that's why it's called Agrobacterium tumor faciens. It causes tumor formation of the plant in those areas where it's infected by this bacteria. Okay, so let's recap the gene delivery methods that we have learned so far. We have learned to use plasmid as a vector to introduce the gene to the bacteria. Just now, we look at using bacteria as a vector to introduce the gene to the plant cell, like this diagram here. Those in blue color are in a syllabus. We have learned the use of retrovirus and adeno-associated virus, AAV, in gene therapy. We have also learned micro-injections in injecting the growth regulating hormone into the embryo, into the fertilized egg of the salmon fish. Right, so all these 4 are the gene delivery method that we have learned so far. So let's look at the genetic engineering of herbicide-resistant plant. Step number 1, obtain the wanted gene. Step number 2, obtain the plasmid. Step number 3, insert the gene to the plasmid to produce recombinant DNA. Step number 4, insert recombinant DNA into bacteria. Step number 5, use the bacteria to infect plant cell, injecting the gene into the plant cell. Step number 6 is called cloning and expression, right? So this is a plant cell, so we grow the plant cell in the lab. So it's called cell culture. So culture the cell or tissue of the plant in the lab using agar medium. Okay, after that, allow the tissue or cell to grow into a new plant, which is called transgenic plant. So the steps involved in GE of herbicide is very similar to the GE of insulin. So you know one, you know all. Okay? Okay, so what is the detrimental effects of the herbicide-resistant soybean? So let's say, for example, in your field, right, you got herbicide-resistant soybean growing there. Can you think of any detrimental effects of this plant to the environment? Okay, see here. Now, this plant can be a super weed. Why? Because you cannot kill it with herbicide. So if we start to grow in another field, say for example, this is a field where you grow your herbicide resistant soybean. So your herbicide resistant soybean. So you grow it in this field. And then somewhere near to your field, you've got another farmer with his own farm here, where he grows, let's say, I don't use the same color, use different color. So let's say nearby, there's this organic farm, okay, organic farm. So let's say, for example, Andrea owns this organic farm, happily growing all the food that is healthy for the body. But too bad, the neighboring field is growing a herb-resistant soybean, which is actually considered as GMO. So this is GMO, beside organic farm. Okay, so we say that, okay, never mind, but the crop plant cannot walk on, right? It'll remain there. But don't forget, pollen can spread. Pollen can spread or not? Yes, pollen can be spread by wind and insects. So the insects may be visiting this farm, and after that fly, go to organic farm. So what happened? Or the wind may blow right this way, and then blow all the pollen to this organic farm. Or the insect can fly to the organic farm. Okay, so and carry with it the pollen from this GMO, and then go and cross, means interbreed with a wild species of soybean in her organic farm. Let's say, for example, she also plant soybean, but it's a different species, right? Because organic. Now, in plant, it's very interesting one. In plant, right, different species can interbreed one. Why? For example, think of orchid. There's so many different types of orchid, right? In terms of size, color, shape, right? So each type of orchid is considered as one species. But because they're orchid, they're very closely related, so they can interbreed. So same for soybean. Soybean, right, got many different species of soybean one. But because they're closely related, so they can breed. So what happened is that, okay, so she's planting soybean, or maybe she's not even planting soybean, never mind, but there's some wild species of soybean here. So when the pollen spread, right, is going to interbreed with the wild species of soybean and produce a new plant, which we call the hybrid offspring. And this new plant will contain that foreign gene. And then after that, when this plant continue to grow and to multiply, then a lot of soybean plants now is carrying that foreign gene already. So we will be contaminating the wild species. Contaminate means we add this inserted gene into the wild species and contaminate the natural wild species. Okay. So what happened when they become contaminated? They also become, oh my God, super weed. Because now they become what? Herbicide resistant. So when they start, let's say when Andrea, suppose organic farm cannot use, let's say someone in a farm start to discover got contaminated already, they want to kill the super weed, right? They spray the herbicide, cannot kill, why? Because they're herbicide resistant. So that's the problem. We have produced super weed and continue to produce super weed by cross-pollination. Okay. Okay, next thing. Now, when farmer use herbicide, any chemicals that we start to introduce to kill an organism, eventually the population of organism become resistant one. You know, when you use antibiotics, right? After some time, the bacteria become resistant. When you use herbicide, after some time, the weed also becomes resistant to herbicide. So whether we produce GMO or we don't produce GMO, the weed will be resistant to herbicide anyway, right? So what's the difference whether we use GMO or not? You don't use GMO, also you use herbicide. So resistant strain of the wheat will be produced also. Okay. The next thing, when we start to spray a lot of herbicide may be harmful to us who consume the food. So this diagram actually illustrate illustrate the cross-pollination of GMO and wild species, leading to all the hybrid of the wild species contaminated with the inserted gene. Okay, so that's for this example. So we have looked at the first example of GMO is the Atlantic salmon that grow very fast. Second example is the herbicide-resistant soybean. Now, third example, which is the last example we're going to look at, is the insect-resistant maize and cotton. So if you look at this picture that I'm showing you here, you will see that on the top, hang on, the top picture, okay, is the pest of cotton. So this is the species name for cotton, Gossypium hirsutum. It's the species name of cotton, okay? Now the pest is what? Bollworm. You see this worm here? Why it's called boll? Because this fruit, right, when it ripened, it will split open and expose the white fibers inside it, which we call it cotton. So that is the white fiber inside it is actually called the boll, right? So the worm will feed on the boll. Okay, so bollworm, which is the caterpillar of the cotton bollworm moth. So it's actually caterpillar of the moth. So when they feed on the boll, right, you know that the yield becomes less because the fibers eaten by the larva and cannot be used anymore. So it reduces the yield. Now, same as on the left side, this diagram shows you what? It shows you another type of moth, not the same type, another type of moth, where the larva of the moth, now caterpillar is larva also, when the larva of the moth actually feed on any part of the corn plant or the maize plant. You can feed on the leaf, you can feed on the stem, you can feed on the kernel, you can feed on the stalk, practically everything. So when it does that, then it will reduce the yield of the maize plant. Okay, so this pest is called European corn borer. Why? Because borer. So what is borer? Borer means to make hole or to drill. So what happens is that the eggs are laid on the leaf. When it hatches, the larva starts to, or the caterpillar starts to feed on the leaf. So as it feeds on the leaf and it's progress, it will start to go to the stalk. So what does it do to the stalk? It will make hole as it feeds on the stalk and make a tunnel. And then when it becomes very, the whole stalk becomes hollow and it's not strong enough to support the entire plant. So the plant will fall down. Then the yield becomes less already. Okay. So when the stalk is hollow, it cannot support the ear. The ear is the part of the plant that carries the corn. So those are the ears that carries the fruits. So that's called the ear. Okay. So the European corn borer also burrow to shank, cobs, and kernels. Now, what is shank? Shank is this one here. Okay. This part that hold the corn onto the stem is called the shank. So you see it start to bore into the shank and they bore into the what? The stem. Can you see that? Making a nice little tunnel. And after that, they go to eat the kernel. So as a result, you can see on the left side, would you want to buy those corn on the left or you want to buy the corn on the right side? Of course, the right side, right? You don't want to buy those on the left, even though the left tells you that this one is pesticide free, you see, okay? But you want nice one, right? So because this one is a BT corn, especially GMO corn, right? This one is non-BT corn. Now, what is BT? You ask me, teacher, what's BT? Now, let's look at here first. Left side is control plant, right side is BT plant. Control plant, you put the moth there, and then they lay eggs, and then their larva starts to feed on the leaf and then die. This one, larva feed and feed, larva die. Leaves are still there. Okay. Okay, so how do we make this plant resistant to insect? So we need to obtain a gene from this bacteria. So the word BT comes from the bacteria Bacillus thuringiensis. So this is where the word BT comes from. Okay, Bacillus thuringiensis. Why BT? Because the gene code for a toxin that is actually synthesized by this bacteria, right? This bacteria, Bacillus thuringiensis, will synthesize Bt toxin when the surrounding environment is in adverse condition. That means when the external environment of this bacteria, which is rod-shaped bacteria, so when this rod-shaped bacteria, right, surrounding environment has got in adverse condition, means lack of nutrients, okay, high in toxic metabolic waste, high competition with other bacteria. So in this kind of condition, it will start to produce Bt toxin, just like how bacteria produce antibiotics. In a similar kind of condition, they produce antibiotics to kill away other bacteria or to stop other bacteria from growing, so that with whatever limited nutrients that they have now, they can still use it to grow. So that's why they produce the antibiotic, and that's why this bacteria produce a toxin. Okay, so this is the toxin is very interesting. It's in the form of crystal one. Okay, hang on. So this Bt toxin is in a form of crystal. You can see here. It's like it's a crystal, it's solid one. So this is structure of the Bt toxin. So this is a crystal that acts as an endotoxin. Endo means inside. Toxin means toxic, right? Endotoxin means this toxin works inside the digestive tract of the larva. Different species of bacteria can produce different toxins that act against different insects. Okay, so Bt bacteria can produce Bt toxin, which is effective against the larva that we mentioned just now. Okay, so we use vector to transfer Bt toxin to the plant cell. So same thing, the vector is Ti plasmid and Agrobacterium tumefaciens. So this is the bacteria which is rod-shaped, and this is the crystal that's synthesized, and also the spore will be synthesized in adverse condition. Now we're going to look at how Bt toxin can cause death from starvation. Now this diagram is a bit blurred, so I'm going to use this diagram on the right side. So this is Bt Bacillus thuringiensis. Obtain the gene for Bt toxin from this bacteria, insert it into the corn and cotton plants. Okay, and then when the larvae or caterpillar of the moth feed on any part of the plant, the cells will be producing, plant cells will be producing Bt toxin crystal. They will consume the Bt toxin crystal. When they go into the digestive tract, enzymes will digest it and make it into smaller molecules that are soluble. So therefore, from original crystal that's insoluble, so it's called protoxin, means before it becomes active toxin. After being digested, it becomes the active toxin. So what does it do? It will bind to a receptor on the membrane of the epithelial cell of the midgut, means the digestive tract. So it binds to the receptor in the epithelial cell of the midgut. To me, it's digestive tract. Okay. So once it binds to it, it forms a pore across the membrane, and this pore allow a lot of ions to pass through it into the cell. So when the ions enter the cell, then the water potential of the cell will drop. Correct? So because of that, water will follow and enter the cell, and the cell will continue to swell, and then it will burst and die. So that's how we make the intestinal epithelial cell die, because of the toxin. And because of that, the organism cannot digest and cannot absorb the food into their body. So basically, they have food in the digestive tract, but they cannot digest and absorb it. So they die because of starvation. Okay. So now you see, now you die. Now you eat it, now you die. Because this is what? This is not normal corn plant. This is BT corn plant. Okay. BT corn plant. Okay. Okay, so how do we use genetic engineering to produce BT corn plant? First, obtain the wanted gene, which is the BT toxin gene from Bacillus thuringiensis. So obtain the wanted gene, and then insert it into the plasmid, which is TI plasmid. Insert TI plasmid to the Agrobacterium tumefaciens, and then infect the callus, infect the cell, infect the tissue of the plant with the Agrobacterium tumefaciens. Now, all these are plant cells. So now after infection, we want to clone and express the gene, right? So this is following the steps for genetic engineering. Now, what is the whole cell that we try to insert the gene to? The whole cell must be plant cell. Okay, it must be, like I said, cotton plant cell in the form of callus, in the form of cell, or in the form of tissue. So just to introduce what I mean. Cell, you know lah, plant cell. Callus is actually, please write, a ball of undifferentiated cells, okay, that can divide and differentiate into a plant. Okay, so this is very similar to our embryo, such as in the form of morula. Now, in animal, after fertilization, zygote divide repeatedly to form a ball of cell. So that ball of cell is called morula. Now, it's the same way. So morula is for our animal, right? Embryo. Callus is for the plant. So this ball of cell very interesting because why? Why they want to grow this callus? They can use special chemical to induce the cell to grow to become a ball of cells, callus. Now this is a ball of cells. Okay. A ball of cells. Now, because all the cells here are undifferentiated, right? We can actually split it into half, and each half can grow into a new plant. Just like the embryo. Why are they identical twins? It's because after fertilization, the morula stage, they split into two. Each one can grow into a complete organism. So therefore, scientists say, okay, why not we produce callus? After that, we can actually cut it into small pieces, right? From one callus, we cut into small pieces, and then we can grow 10 plants out of one callus. Correct? So they want to propagate the plant, we're going to multiply the plant faster, so they use this method. So they use callus. They grow callus first. Then after that, they cut the small pieces, and then they continue to grow them and make them into new plants. Now, tissue culture means I can grow small piece of leaf. So I take the leaf and I cut it into small pieces, right? Take a leaf and then cut it into small pieces, and each piece is a tissue, right? Each piece is a tissue. And each tissue I can grow into one new plant. So isn't it good? From one leaf, I can grow into many plants. So this is called tissue culture. So you've got callus culture, tissue culture, cell culture. Okay, to clone and express the gene. So then you get your transgenic plant, which can produce Bt toxin. Okay, so now we can produce Bt toxin. Now we can produce Bt plant. We can introduce and grow in the wild. But how will it affect the environment? Because we don't want to simply introduce something to the wild and start to cause havoc to the whole environment and our evolution, right? You don't want to do that. So we need to actually find out first, consider all these things first. So yes, whatever chemical we introduce, eventually insect pests become resistant, right? Okay, so if after we grow the plant for a long time, suddenly there's this, hey, how come this plant got European corn borer and bore all the corn, what happened? That means to say that resistance had developed in the European corn borer. They developed resistance towards the Bt toxin. Okay, then after that, our GMO is no longer useful for us already. Okay, because it cannot kill the insect anymore. Okay, non-targeted insects may die. Okay, see what happened? Plant produce pollen, right? So even though the insects do not feed on the plant, but once the pollen spread out, pollen is actually food for a lot of insects. So when they feed on insects, they die. So they are the non-targeted insects, but they die because they feed on the pollen. Okay. And also the pollination can transfer the gene to other species which are close relative, and then it can create superweed. Superweed, yeah, correct. Okay, superweed, because why? Insect cannot kill them. So they become superweed. Okay. But the scientist says this is not an issue because pollen can only survive 2 hours. After the pollen starts to spread away from the flower, right? It's away from the body of the flower of the plant already. So it cannot survive for very long. So it can only survive for 2 hours. Whatever nutrients they need is only coming from the pollen itself. So it cannot survive very long. Okay. So that will deter the worries that, oh, they can fertilize other plant and then create a superweed hybrid. Okay. So advantage of insect-resistant cotton plant. Because it's insect resistant, right? We don't have to spray insecticide. So it will prevent us from killing the non-targeted insects, because you spray the whole field, right? Then all the insects will die. But now only insect that feed on the plant will die. Then we will not kill all the non-targeted organisms. This advantage, okay? And we only kill the insects feeding on the crop. So now we want to see disadvantage. There will be possible detrimental effects on the environment, which I will show you afterwards. There'll be possible loss of biodiversity because we keep on growing insect-resistant cotton, right? So the non-resistant type, the normal type in the farm, we no longer grow anymore. After that, they'll be lost totally, from Earth. So all the genetic material that belongs to them is no longer available on Earth already. So when we need their genes, we don't have it already, because we don't grow them anymore. Okay. And then GM seeds are very expensive because they're produced by genetic technology, right? They're expensive. So you want to use GM seeds to grow GM crop, and then the products will be very expensive. Now this one I'll skip. I will not explain because it's not in the syllabus. Okay, I'll skip this one. I'll skip all of them. Okay. So let's recap. So in order to improve the quality and the yield of farm animals and crop plants, scientists have used GMO to create fast-growing GM salmon, which grow the whole year, herbicide-resistant soybean, which can produce the 3 amino acids and protein, even though we spray the herbicide, glyphosate, and also we can produce insect-resistant cotton, so that when the boll worm starts to feed on the cotton, it will die. Or when the European corn borer starts to feed on the plant, it will die. So very powerful tool to improve the quality and the yield of farm animals and crop plants. But we need to consider, now this is a summary for the 3 GMO, the wanted characteristic, the wanted gene to introduce the characteristic, source of the gene, and GE vector. I'll fill in this one after the lesson for the salmon. Okay, you see the trend in the whole world is, you can see that as the year passes by, the percentage of the planted area with the GMO is actually increasing. In fact, you see the soybean almost go to 100% already. If 2008 is already 90 over, by this time, it should be almost 100% soybean that you can find in the market. They are all GMO, unless specified organic. That's why organic soybean, they are very expensive, because the supply is less and demand is more, then become expensive. So if the trend is there are more and more GMO being cultivated, That means to say we have to really check the ethical consideration and social consideration of introducing them to our environment. How will it affect us? How will it affect other organisms? Okay. So let's summarize the social and ethical implications of using GMO. We may start to produce super wheat that's resistant to insect, resistant to herbicide, resistant to drought. We may potentially contaminate wild relatives and conventional crop plants by cross-pollination, producing hybrid offspring. So that new gene escape to the wild, we will start to change genotype and phenotype of the wild plants, and we will lead to evolution. We don't want to play God. That's the thing. Because what is the outcome? We do not know. Okay, so we don't want to do that. It must not pass to the offspring. So that means if you're a farmer, right, you grow your crop plant already this season, right? You have to go and take new seeds from the scientists and grow again. Because we don't want them to pass to the offspring. So we have to take new seeds, okay? Promote development of herbicide-resistant insect. Yes, the moment we use more herbicide, resistant insect will grow. Even the plant can produce herbicide, resistant insect will grow also. And those chemicals, remember the Bt toxin in the plant. If the insect feed and die, then we go and eat the plant, we eat the corn. So what is the effect of Bt toxin when we eat it? Is there any side effect? Is it toxic? Can it cause allergy or not? We have to test all this first, right? Okay. Herbicide used on crop leaves toxic residues in the crop. This one is whether you use GMO or not. The moment you spray herbicides, pesticides, the residues of the herbicides will be found in the plant. If we feed on it, we will also eat on those toxic chemicals. So what is the effect on our body? Okay. GM seeds are expensive. So let's say if GM seeds are expensive and the price of it is similar to herbicide, why not you just plant normal plant and spray herbicide? Less risk, isn't it? Okay. Less risk of contamination and playing God. Growers need to buy seeds each season, so it's very high cost. Some grower kept seed from one crop to sow for the next. Yeah, let's say, for example, you can keep some of the seeds just in case they have got the introduced gene, you can continue to grow the GMO. Okay. Potential losing of biodiversity. This is very important because we keep on growing the GMO, like a lot of fields start to grow a lot of GMO, and then the traditional varieties are not grown anymore. After a long time, you'll find that all these plants are no longer existing on Earth, so we will lose their background genes. Background genes are genes in the plant that's needed to support them to grow in a particular location as well as climate. Okay, that means to say that if you're planting a rice plant, how come the rice plant can only found in countries that near to equator? It's because they got the background genes to allow them to survive at, let's say, high temperature, high rainfall, okay, submerged under the water. So these are all coded by genes, right? So if you don't plant this kind of rice plant, you say, yeah, this one very tedious. Why not plant the rice plant that can grow in the air one? Okay, hydroponic, right? And then after a long time, you lose all these background genes already. Next time. Okay, so when the climate start to change, when climate start to change, and then we need some of the genes that allow the plant to grow at maybe very high temperature, we cannot find those genes anymore. Okay, so solution, we have to use seed bank. Even though we don't grow the plant, we keep all the genetic material in the form of seed in the seed bank. That one is solution. Okay, safety effects on environment and health not sufficiently tested. So just now we said that potential risk of the chemicals if you consume, if the animal consume, please test it. Okay, so they said not sufficiently tested because of that, not sufficient information to show whether there's the gene can escape, whether superweed has been formed, whether the chemical is toxic and allergenic to organisms that eat them. Okay, so who have got the intellectual property rights to GMO? So if you create GMO, do you have the right to GMO? If the doctor create the test tube baby, who have the right to all the embryos that are not used, they're discarded? Can you do research on the embryos? Who have the right? Okay. So some say, why not don't create GMO? We just, in agriculture, we aim for a balanced diet and fresh vegetables. So instead of planting GMO crop like corn, why not you plant something that can also provide you with protein and carbohydrate? Okay. This one actually applies to golden rice, which I do not teach you in your syllabus, sorry. So this part here, this one is for golden rice. So you can ignore this one, okay? Because golden rice is no longer in your syllabus. This is for golden rice, okay? So next, are GMO food labeled or not? So in the supermarket, you go, right, whatever GM food must be labeled so that people can choose, because they do not know the risk of GM. So therefore, they may not want to eat GM anyway. So you have to label it properly. Many possible effects cannot yet be measured. Because what are the effects we do not know yet? Only the time can show us. Okay, so these are all the ethical and social consideration of introducing GMO to the earth, to our society. So what you have learned in this lesson is using genetic engineering to improve the quality and productivity of farm animals and crop plants, example, GM salmon, herbicide-resistant soybean, and insect-resistant cotton. Then we also look at the ethical and social implications of using GMO in the food production. So that's all for today, and we have completed the chapter for gene technology, except for one topic, which is gene editing, which I've not taught you. Okay, so I'll teach you gene editing. Then we will be starting a new chapter. Okay, so that's all for